Alzheimer's disease is defined by the Merriam-Webster Dictionary as a degenerative brain disease of unknown cause that is the most common form of dementia, that usually starts in late middle age or in old age, that results in progressive memory loss, impaired thinking, disorientation, and changes in personality and mood. Alzheimer's disease can be caused by both environmental and genetic factors. Variations in genes can either lead to a person having risk genes or deterministic genes. About 40 to 65% of Alzheimer's patients have risk genes that increase their risk of developing Alzheimer's. These genes do not mean that a person will undoubtedly develop Alzheimer's. They only mean that a patient is more at risk for developing the disease. Less than 1% of patients have deterministic genes. These genes guarantee a patient will develop Alzheimer's. Additionally, Alzheimer's patients do not need to test positive for any risk or deterministic genes to develop the disease. Familial inheritance only makes an individual more likely to have the disease. There are 4 genes which cause an increased risk of developing Alzheimer's: amyloid precursor protein, presenilin 1, presenilin 2, and apolipoprotein EE4. Variations in the PS1 gene are the most common cause of inherited Alzheimer's, while variations in the APOE4 gene are the risk gene that causes the greatest impact. Variations in all of these genes affect the processing or production of beta amyloid protein. This protein is one of the main components of plaques.
For our experiment, we're going to be studying 3 different SNPs. First is RS429358, located on the APOE gene. The APOE gene encodes a protein involved in metabolism of fats in the human body. The CT genotype causes 3 times increased risk of Alzheimer's disease, while the TT genotype is the most common. Next is RS1459994145, located on the PLD3 gene on chromosome 19. This gene normally encodes for member 3 of the phospholipase family of enzymes which catalyze the hydrolysis of membrane phospholipids. The AA and AG genotypes both double risk for Alzheimer's disease, while the GG genotype is most common. Lastly is RS7593268, a SNP on the TREM2 gene on chromosome 6, which results in a substitution. TREM2 encodes a membrane protein called a triggering receptor expressed in myeloid cells. CT genotype triples risk of Alzheimer's disease, and the TT genotype increases risk of Alzheimer's by 3.5 times. CC is the most common genotype.
We selected which SNPs to test for based off previous research that linked them to Alzheimer's disease. A 2011 study showed that the C variant of RS429358 was found in 23% of Alzheimer's patients tested. A 2014 study concluded that the A variant of RS145999-145 doubled chances of developing Alzheimer's. A 2009 study showed that the T variant of RS7593268 can over triple the likelihood of developing Alzheimer's. And even though this variant only existed in 0.5% of the study group, we still decided to include it in our test because the risk is so high.
So what are the drawbacks of taking this genetic test? While coming back positive for the APOE4 gene, the TREM2 gene, and the PLD3 gene does increase your risk of Alzheimer's, it does not mean you will 100% develop Alzheimer's. Carriers of a single APOE4 gene have a 2 to 3 times increased relative risk, yet this variant still has a low predictive rate. Additionally, a large factor of whether you develop Alzheimer's is not based on genetics but on environmental factors such as smoking, head trauma, and age. Finally, if you do get this genetic test and you come back for being at a higher risk of Alzheimer's, what next? Unfortunately, at this time, your options are limited due to there being no cure for Alzheimer's. For all these reasons and the pricey cost of genetic testing, it may not be in your best interest to get this genetic test.
Since there is no known cure for Alzheimer's, many medical professionals tell patients to stay away from testing due to the fact that early knowledge of the disease will not help from a medical standpoint. Additionally, it is very easy to jump to conclusions if a patient tests positive for some Alzheimer's risk genes in an at-home test, but the appearance of these genes does not necessarily mean one will develop Alzheimer's. However, since the test only costs about $100 to $200, it does not put a large financial burden on patients.